let's switch gears and talk about targeted proteomics. I'd like to basically just introduce the basic concepts and essentials about targeted proteomics today. So far, what you've heard was about discovery proteomics, where we are trying to sequence as many peptides as we can. We're shooting many, many arrows in our sample. And that's actually the approach that we take in any study, or most of the studies that we start, at least in the beginning step of the study. And it's a great way to start the study. It can be relatively quantitative if labeling approaches are used, just like Jake just introduced it, either metabolic or chemical labeling approaches. However, there is one issue with discovery proteomics, and that is that you don't know what you're going to hit, and you don't measure each and every peptide every time. So sometimes when you run the same sample twice, you may not hit the exact same peptides in the second, and that's due to the stochastic nature of sampling that happens during the discovery experiments. So however, if you, let's say you have a biological system where you already have information about certain proteins that you want to measure each and every time, or after your discovery experiments, you generate list of proteins that you are interested in and you want to go and probe that in many more samples. This is where you will use targeted mass spectrometry to look at fewer peptides or proteins, but try to detect and quantify them at each and every time. And here, those peptides and proteins will be defined by the user beforehand. And the quantitation can be highly precise if internal standards are being used. And this is a more sensitive approach than the discovery proteomics. And this chart just kind of another way of comparing both approaches together. A lot of times discovery proteomics we also call shotgun proteomics. You can see that in discovery proteomics we can actually sequence many, many thousands of peptides, whereas in targeted proteomics the list can be tens or hundreds of peptides. But targeted proteomics is much more reproducible and sensitive, because now we're targeting the peptides or proteins of interest as opposed to looking at everything else. And this chart kind of nicely shows the proteome pool coverage, where if you are doing discovery proteomics, you are going to start by detecting the abundant stuff in the sample first, and then go as deep as possible, also detecting the abundant stuff. Whereas here, you're avoiding the abundant stuff if you're not interested in them, and just already trying to dig deep into your sample. And of course, the targeted proteomics using the latest generation high-tech instruments, you can get 5 orders of magnitude dynamic range. So when you look at the sample, so let's say you have a complex sample which you ionize into the mass spectrometer, and if you try to scan out everything in your sample, you will get a signal that looks like something like this, that where you are detecting all of the peptides in your sample, scanning out everything and detecting all of them in the mass spectrometer. Pretty complex trace right here. So if you do a selection, apply first level of selecting, first level of filtering to in the mass spectrometer, where you just select to look at certain, either one or number of precursors or parent ions or peptides, you will get simplified signals, so simplified spectrum or chromatogram. But again, there's a lot of noise. When you apply the second level of filtering, this is where you will select specific fragment ions coming from a specific precursor of your interest. You will see a signal like this, much, much cleaner. And at the same time, we can actually not only look at one of these, but hundreds of them together. So this is the approach that we take in targeted proteomics, and we can actually measure and quantify simultaneously tens to hundreds of peptides, and this approach is most sensitive approach. It's 50 to 100 times sometimes more sensitive than discovery proteomics. So before we go any further, I'd like to introduce some terminology here, because we're going to use these abbreviations a lot. So selected reaction monitoring or multiple reaction monitoring, in other words, SRM or MRM, are the two acronyms used a lot, but both of them really refer to the same mass spec method. Selected reaction monitoring is the singular form, multiple reaction monitoring is the plural form of the method. We're going to talk a lot about transitions, and transition is really the parent ion coupled to a specific fragment ion, another way of saying Q1, Q2 pair. We're also going to talk a lot about peak area ratio. This is usually the light peak area ratio to the heavy peak area, and many of these Jake already talked about before. So we do MRM type experiments in triple quadrupole mass spectrometers. And triple quadrupole mass spectrometers have 3 quadrupoles, where first and third quadrupoles are scanning quadrupoles. And the second one is the collision cell. This is where all the fragmentation happens. So when we do data-dependent acquisition and try to collect full-scan MSIMS, when we spray the complex sample into a triple quadrupole mass spectrometer, in the Q1, we select specific one peptide at a time, and then we fragment that in the collision cell and then scan out all of the fragments that belong to this particular peptide that we selected out to the detector. And what we get is a full scan MSIMS, which includes all of the ions that you heard already, B ions and Y ions in it. When we do MRM-MS, what happens is that when we, again, spray this complex sample, we apply the first level of filtering where we select the parent ion, peptide precursor, then fragment that in the collision cell, and after that we select a few 3 to 5 transition ions, fragment ions that are specific to that precursor ion, and only detect those selected fragment ions that are called transition ions. And that's why this is highly selective, because we're applying 2 levels of filtering here. Again, going through the benefits of MRM-MS one more time, it has high molecular specificity. The sensitivity, we get great sensitivity with this approach, and assay performance. And we can detect sub-picogram per mil levels, and the assay performance is sufficient up to microgram per mil levels. This MRM-MS allows us to detect and avoid interferences, and I'm going to talk about them more later. It does not require any antibody, immunoassay grade antibodies. This assay is highly multi-flexible, so we can measure hundreds at the time, and it has high inter and intra reproducibility. And as I said, now using the latest generation mass specs, we can detect down to low, mid picogram per mil range, but improvements in mass spectrometry will only translate into better MRM assays. So how do we construct MRM assays? This flowchart shows the process step by step, and I'll go over them one by one. So first, we start with the protein of interest and design define the signature peptides that are going to act as surrogates for quantitating these proteins. And then we synthesize the heavy labeled internal standards for these peptides and configure MRM assays and apply them to the samples to measure. So when we apply it to the samples, we spike the heavy labeled internal standard peptides into the sample, which already has the endogenous light peptides. And then both of them together that are in one sample are sprayed into the mass spectrometer, and we detect light and heavy pairs and then use the ratio to do the quantitation. So first step of a process is defining the signature peptides. And we usually, the first approach we take is our experimental data. If we have any data that exists in-house, of course, we use that for the particular protein of interest to select peptides for the quantitative assay. Or if none exist, we always use any data that's available in public repositories. We sometimes supplement this by the predictive algorithms. If Or if the experimental data does not exist at all, we just have to use the predictive algorithms to pick these peptides. And some of the criteria for selecting these peptides are listed here. These are all, of course, preferences. Sometimes we don't have a luxury of following all of these preferences. The first one is actually, it's very important that the peptide has to be unique to the protein of interest. And we try to choose peptides that are 8 to 21 amino acid long for practical purposes, not too short or not too long, that are going to be problems afterwards for chromatography. Try to avoid methionines or cysteines and glutamines at the end terminus for the reasons that were already mentioned here today. And again, with dibasic amino acids at the ends, N terminus and C terminus, again, to get better digestion or better reproducibility of them. Second step of the process, once we have then of course, once we choose the peptides, we then synthesize, order those peptides in heavy labeled forms. Again, using the C13 isotope, just like Jake explained a little bit before, using the heavy labeled C13 isotopes in one of the amino acids, synthesize those peptides. And once we have the peptides, then we configure the MRM assays. And these configuring assays now consist of choosing specific transitions for each one of the peptides of interest. And usually those are, again, few preferences for choosing them. We try to choose Y ions and singly charged Y ions that are the MZ of which are bigger than the precursor mass. And then we optimize mass spec parameters for these transitions to get maximum transmission of those transitions. So this is an example. Let's say we've selected this peptide and got the peptide in heavy labeled form. When we spray this peptide into the mass spectrometer and fragment it, this is the full scan MSIMS that we're going to get. You can see all of the different fragment ions here. We choose these 3 fragment ions, Y6, Y7, and Y8, for the transitions to move to the next step of our MRM assay configuration. We choose them because they are very abundant. They are larger than the precursor mass to increase the specificity of the assay. And so when we do MRM-MS assays, of course, as I already said, we use the heavy labeled internal standards. And then the final method is going to include the transitions, the same transitions for the heavy peptide and the light peptide, which is our endogenous analyte. Here you can see 2 different views of each one of them. The stick, what we call a stick view or spectrum view, of showing each one of the transitions, as well as the XICs, extracted ion chromatograms of the 3 transitions for the light peptide. And on the bottom, it's the same view for the heavy peptide. So what's important here to mention is that essentially the heavy and light versions, like Jake already said, are the same peptide. And the only difference in between them is the mass. And that's how we distinguish them in the MRM-MS assay. So the mass, the MZ, is different at the parent level. Also, the fragment ions, transition masses, are different. So that's how these XICs are unique. But because it's the same peptide, they all coelute. So we get these XICs at the same time in the chromatogram. And that's how we actually detect that this is the light version and heavy version and quantify them together. And this is a very important phenomena because this is what actually helps us in identifying, or actually more like detecting and avoiding interferences. The transition ratio among the 3 transitions that we're monitoring is extremely important. And that's what we actually use as a standard to look for interferences. So for example, in this case, we are actually monitoring 4 different transitions for a particular precursor ion. And here it's plotted about 3 or 4 minutes of this chromatogram, where we are detecting or monitoring 4 transitions. And you can see there is number of peaks here. Now, we need to know which one is the peptide of our interest. Because why we have number of peaks? Because most of the time, our sample is very complex. So there's interferences a lot in the sample. And interferences are going to, they become really issue when they coalute with our analyte. So really, there's like 1, 2, or 3 peaks that have all 4 transitions present, but at different relative ratio of the transitions. But because we have the heavy standard, we know that this is the correct spectrum for our analyte to monitor and to quantify. Here, and again, another slide showing very nicely the interference. This is a study which we did like years and years ago, where we had PSA spiked in plasma and then fractionated by strong cation exchange and quantified by MRM-MS. And here, on this panel, it's the fraction 1, and on the right side panel, it's fraction 2. On the top panel, we are seeing the internal standard and the relative ratio of the transitions from internal standard, how it nicely behaves. The green one is the most abundant, and blue and red are like equal almost. And we can see that this is the analyte, the XIC view of the analyte, and on the bottom is the spectrum view of the analyte. So you can see that in fraction 1, the transition ratio of the analyte very nicely matches to the transition ratio of the internal standard. Whereas on the right side, on the left, on the right side, the transition ratio does not match at all because this green transition is interfered by another species that coalutes with our analyte of interest. We actually were even able to go ahead and try to sequence what this is, and we were able to partially sequence this other peptide that happened to coalute in this one of the fractions. So interferences can be a big issue in MRM-MS, and it's very important to detect and avoid the interferences. Not every time to identify the interferences, but at least be able to avoid. And this slide just shows a few examples of interferences in a study where we were looking at calibration curves of spiked proteins into plasma. And on A and B panels, we can see these are clear examples of interferences because only one of the transition does not align with the other 2 transitions, whereas the other 2 aligns very nicely with the expected line. So this shows clear interference in these transitions. Here, in this one, we're seeing actually detecting endogenous levels at the low end because all 3 transitions are above the expected line. There is also another issue that we may have in MRM-MS analysis where it would be more like related to the whole performance of the LC-MS system or not recovering peptide at all. Or in this particular case, it's more like saturating detector, so the most abundant transitions are not actually aligned, but more like the getting saturation point. So this is all is important to actually detect and avoid. And years ago, we used to do this manually. And in fact, this is a very tedious process. In fact, there was a study where it took us 2 weeks for 3 of us to go over data from 7 different sites, and that was only 10 peptides that we were looking. And right now, our multiplexing capability is much higher. We're looking at hundreds of peptides. So it's impossible to do this manually, identify or detect and avoid the interferences manually. So in our lab, we've developed the software, this software audit called audit, which actually automatically looks at the ratios of the relative ratios of transitions of the internal standard and analyte, compares them together by statistical tests, and uses that information along with the percent CVs from the triplicate analysis to flag certain transitions so that we can go ahead and avoid them in our further data analysis. Okay, so once we know this part, a lot about this part of the, or we've configured, chose the transitions for our MRM-MS, the next step is usually we run the response curves where we spike in light and heavy versions of the peptides into relevant background, and this allows us to define limits of detection and quantification, the linear range of the assay, as well as precision and recovery of our analyte. And usually, and this slide just illustrates how we do it. So again, this has to be done in the relevant background where we spike in heavy peptides at constant concentration and vary the light peptide amount. And then all of these samples are analyzed by triple quad mass spectrometer by MRM-MS. And what we get is that XICs of heavy and light peptides, and you can see light illustrated in red. That will go up, of course. And then we use this in looking at the linear range, plotting the calibration curve and looking at the linear range. We use the lowest concentration points for defining the LOD and LOQ of our assay, as well as using this information, we also look at the reproducibility of our assay. And again, this is done all automatically now due to the software that we built within the group called Quasar, which you will hear a lot about tomorrow by Mani and Sue. So what are some of our LODs and LOQs in these MRM-MS assays? Of course, that depends from the background or the sample, how complex the sample is. And also, it depends how much we want to process, or we have the luxury of processing our sample. If we just take our sample, digest, and add the heavy labeled internal standard and measure, we're going to look at the most abundant proteins only that are in more like microgram per mil levels. If we employ some depletion strategies where we deplete most abundant proteins, and then follow that by digestion and adding internal standards, and we can actually get into more like 100 nanogram per mil range. If we employ another level of fractionation after we generated peptides, or we use antibodies, protein antibodies, or peptide antibodies after protein digestion, we will get down to picogram or nanogram levels of detection in MRM-MS assays. Okay, MRM-MS is, it provides precise relative quantification. Okay, as it's been said a lot today, we always digest our proteins. So let's say we have a protein of interest, we digest into peptides, and usually what I haven't said so far, when we design the MRM-MS assays, we usually target 3 to 5 peptides initially. We try to start more because at the end we may actually lose some after throughout our MRM assay optimization. But we hope to get at least 2 peptides, at least 2 assays configured at the end of the day. So let's say we digest our protein into peptides, then add 2 heavy labeled peptides, and then measure them by MRM-MS. There's times when we will get differential recovery of these peptides, and that's illustrated by these 2 panels here. So this is peptide 1, this is actually CRP recovered, CRP digested, and the 2 peptides belonging to CRP. And you can see this is the blue is always internal standard, and red is the light peptide. The peak area ratios are very different. They're like about more than 2 times different in between 2 peptides. So we are recovering one of the peptides much better after the digestion than the other one. And this can be the case a lot. So even though we are precisely quantifying these peptides, but our recovery is different for each one of these peptides. What can we do to improve this even better and go one step closer to the accurate quantitation? Would be to use labeled protein standards. So here, in this assay, we had both. We had heavy labeled internal standard peptides in blue, and we had N15 labeled protein standards in brown. So you can see in one assay, we're measuring actually 3 forms of the peptide: light version, heavy peptide, and the peptide that came from the heavy protein. And you can see on the left panel, when the light peptide is ratioed to the heavy labeled internal standard peptide, you can see that the line is below the expected line. And we are getting, if we just pull one of the concentration point, we're getting about 50% recovery of that peptide. However, when we ratio the light to the protein internal standard, actually it nicely aligns with the expected line now, and if the same concentration point now shows around 100% recovery. Again, illustrating that using protein standards, we will get one step closer to accurate quantification. But not every time we'll have the luxury of using the protein standards, so everything is a cost issue and like considerations when developing MRM assays. So MRM-MS assays are now being widely used in proteomics and being applied to various different studies in proteomics. So here, you can see in this slide taken from this, the Nature Biotech paper by Ebersol's lab, it's nicely illustrating all the different areas where MRM-MS is being employed. For example, it can be used to look at metabolic pathways. So in cells that undergo certain perturbations, you can actually digest the proteins, add the heavy labeled internal standard peptides, and measure the changes. Or you can look at the stoichiometry of different proteins in protein complexes. If you just immunoprecipitate your protein complex of interest, and again, spike with heavy labeled internal standard peptides, you can get some information or be able to look at the stoichiometry of these proteins in the complex. Or you can use MRM-MS to look at the signaling pathways by quantifying phosphopeptides. Here, you will need to synthesize heavy labeled phosphopeptides, and then do some kind of, after the digestion, do some kind of enrichment to look and quantify the phospholipeptides based on the internal standards. Or you can also look by using this MRM-MS at the different modification states of the same protein and look at the modification patterns. And this is being done within our lab already in histones. And finally, MRM has been used widely over the last 6-7 years in biomarker discovery type experiments or verification, where you have some candidates and you want to look at the candidates and measure them in many more samples. You will employ MRM strategy and measure them in different populations. And with this, there's like a number of references for you to look at afterwards. Some of them are from our lab, and some are from outside. This concludes the day, right?